and welcome to the Jones Seminar. It's my pleasure today to very quietly announce the Jones Seminar for this week. We're very fortunate to have a sabbatical visitor here at Dartmouth this year from Caltech. His name is Axel Scherer, and he's the Bernard Netjes Professor of Electrical Engineering, Applied Physics, and Physics at Caltech. He's also the co-director of the Kavli Institute in nanoscience. His research focuses on the development and application of new fabrication and design methods. In the 1980s, Professor Scherer pioneered the development of vertical cavity lasers, which have become a commercial success. In the 1990s, his group developed some of the first silicon photonic circuits, optical nanocavities, and integrated microfluidic devices. Fundamentally new structures such as photonic bandgap geometries resulted in some of the world's smallest lasers, modulators, and waveguides. At the moment, he's interested in miniaturization and integration of microfluidic, magnetic, and optical devices for applications in nanobiotechnology. His group also explores the limits of lithography at the nanometer scale. He has co-authored over 300 publications and holds over 60 patents in nanofabrication-related areas. Please welcome Axel Scherer.
First of all, I'd like to thank you for coming here and taking this opportunity. I'd like to sort of try to reach out to people here to try to build up some collaborations. We've been doing that over the last year with Elsa and others. And I think that now is a good opportunity, since I'm going to be here for a little bit longer, to talk about opportunities of working together. So what I'll talk about today is some of the work that we've been doing on biomedical devices. But what I'll also weave into this is work that we've done on building the small structures that we have the facilities to build in my group at Caltech. The kinds of things that we'll talk about are integrated devices, photonics, fluidics, electronics are added together to build structures for the medical applications. And then at the end of the talk, I'll sort of show you a vision that's not happened yet, that's on its way, of building very small integrated structures that allow us to implant systems into the body to provide us with diagnostics, real-time diagnostics, rather than having to take samples out and analyze them. Everything that we do is fueled by students in my group. I get to talk about these things, but the real people that get to work on structures like this, we build our own vacuum systems and etching tools and so on, are the students. And of course, we get a lot of support for the students from various government agencies. I thought I'd start with that because it's really the enabling of this technology. What we're really trying to do in my lab is to follow the lead, the example of the microelectronics industry, which has over the last 50 or so years gone from discrete devices to integrated chips. And in fact, the same kind of technology that has made this jump possible is now available to do other things. And in fact, the photonics world is also going into printed photonic systems. And the fluidics world is going into printed fluidic systems. And all of this printing has a long history, and we all know that that printing has made the availability of things like books very inexpensive. And what we can do is now make these other systems, the photonics, the electronic, and the fluidic systems, equally inexpensive by printing them. So the idea is to do what was done back in the Middle Ages, just to do it at a higher resolution. And that leads us to the concept of what Gordon Moore predicted as for Moore's law, this geometric increase in complexity in his case in the electronics world. Now, we should notice that this is a very interesting linear projection that that's still valid up to today, over 40 years. And the one technology, the one device that was used back in the 1960s, started in the 1960s, is still the same device that's really pushing this technology forward, and that's the MOSFET, the CMOS MOSFET. And it's very interesting that this has this great great trend towards complexity and more devices per chip. And there's a very good reason for this. There are actually two reasons. The first reason is that there's a certain amount of money that you have to pay to build a silicon chip. And that's about $5 per square centimeter. And it has been $5 per square centimeter for the last 40 or so years. What's interesting is that the more you can fit into this, the more transistors you can build in that square centimeter, the more valuable your chip, and so that pushes people to add more complexity. There's another thing that forces people to make more complex integrated circuits in electronics, and that's that communication, moving information from one chip to another, is quite expensive. And so the idea is that the power dissipation and all those things of moving information from one place, from one chip to the other, makes it difficult enough that you'd want to do as many things as possible on the same chip. And in fact, there's a great analogy, I believe, for biologists. If you want to analyze a single cell and you want to do this test on the single cell, look at maybe the RNA library or something like that, that's what we sometimes do, you don't want to lose molecules. You may only have about 400 molecules that you're interested in, and you don't want to lose them. So the last thing you want to do is pipette these solutions from chip to chip. You'd really want to keep everything on the same chip, and therefore the same trend that made electronics try to increase the functionality of their chips, I think is going to drive fluidics and photonics to do the same thing. So I'll talk about the integration of optics and electronics and fluidics. And the one thing I'd like you to remember is if we lithographically fabricate electronics, which is already done, photonics, and fluidics, we can make alignment systems, we can make alignment marks that allow us very easily to put one on top of another and align them quite accurately. And so you can integrate these different material systems by using lithographic techniques. And so we'll talk about all of these different applications and our opportunities. So first, let's talk about silicon. Silicon is where we started, of course, with the electronics world. You can make silicon chips in big factories, in fab lines that cost upwards of $1.5 billion nowadays to make. But the infrastructure is there because the electronics industry demands it. So what you can do is you can take one of these fab lines and you can tell them, let's do a different mask set. And you can, instead of making transistors, you can build waveguides. And if you do that, you can build a system where there's electronics and photonics devices right next to each other. You can do that if you don't lose the light. You have to have a special structure called silicon on insulator and so on. But you can build optical switches next to electronic circuits. And that's led us to many different applications in the communications world. Well, in the medical world, what you can do is you can take these kinds of devices and you can change them in function. So here, for example, we have an optical resonator. It's simply a ring of silicon that's sitting on top of silicon dioxide. Light gets coupled into this ring through a waveguide. And at a specific wavelength, the light that goes into this waveguide gets coupled into the ring, and you can measure it out on another waveguide, another port. So it's a filter. It's an optical filter. And what's interesting is you can put something on top of this ring, some chemical, that allows you to now sense whether there's a reaction taking place that changes the refractive index of whatever's on top of this surface. And that allows you to build a little sensor using essentially the same thing that you would use for a filter in the telecommunications industry. So these kinds of rings can have different shapes. You can have a solid ring of silicon, or you can have a slotted ring of silicon, and different reasons to make these different shapes. But what you can do is you can look at the resonance frequency of this, and you can observe it as a reaction occurs. And so initially, the resonance frequency of this ring was at this wavelength, at 1563. And as the reaction occurred, in this case an oxidation reaction, you see the refractive index change. And as a result of that, the length, the optical path length of light changed. It became less, and actually the resonance moved to a different wavelength. And of course, you can measure this if you have an optical spectrum analyzer or something like that. You can also measure it if you have a simple laser. If you take a laser that works at one of these wavelengths or close to one of these wavelengths, you can observe the resonance peak of this ring with respect to the very sharp wavelength spectrum of the laser. And by looking at where that is, where the resonance of this ring is with respect to the laser frequency, you can change the intensity of the light that comes out the other end. And so you can build a very simple spectrometer where you don't have to build a system that includes the entire spectrometer part. But you simply use the fact that you have different spectral purities of these two devices. And you can build a system where you have many different sensors, in this case resonators, that can be observed at the same time. And you look at whichever one changes in intensity. So you can build a system which tells you whether something's reacted or not on the surface. You can build these kinds of systems by functionalizing different rings with different materials. And different materials then react with different chemistries, and you can observe which chemistries react. And the trick really is that you have these rings, these optical structures, which are about 5 microns in size, and you'd like to deposit the chemistry within that sort of resolution, and it turns out we can. We could even make these structures even smaller. We can build resonators that look something like this, where you can basically get a Q of above 60,000 out of a very small waveguide which has been perforated with holes. So we built these kinds of systems and make refractive index sensors. Now, if you want to really have a nice refractive index sensor, for example, this one, have these holes in there. If you build this with perfection, maybe you can go and make a resonator with a very, very narrow spectral line width. And that would then allow you to measure very small changes in refractive index. And that's the goal there. Now, it turns out that you can't make these structures more perfect than what the lithography allows you to do. And so you always are limited, in this case, to Qs of maybe about a couple of 100,000. If you want to go beyond that, there's a trick you can play. You can simply say, rather than using a material that simply is a resonator, we can build a resonator with some gain in it. We can put some material in it that actually adds photons, so we don't just filter, but we also add photons at the same time. And of course, if you build a laser, that sort of does that. And that's what we've done here. We have, in this case, 3 different laser cavities, each one which has a tiny little hole in the center of it. And I'll show you in a moment that the field intensity is optimized to be maximum in that hole. And these 3 lasers emit at different wavelengths. In this case, 3 different wavelengths separated by about 100 nanometers in spectrum. They're only separated by about 4 microns in geometric distance. And if you dip this whole system into a solution, you can observe the refractive index of that solution by simply looking at what the laser lasers at. So in this case, in air, the laser lasers at this wavelength. If you fill this up with some solution, with methanol, for example, You redshift the laser emission frequency, and you get the light emission at a longer wavelength. And if you put it in isopropyl alcohol, it goes even further. And you can measure the refractive index of whatever you have in this volume of about 10 to the minus 17 liters with an accuracy of 1 part in 10 to the 4. You can even put material in this, like, for example, little particles. You can have little beads that diffuse in and out of this laser cavity. This is what the mode pattern looks like. And as these beads move in and out, you actually change the frequency of this laser, and it moves back and forth. So you can measure, you can actually count the number of beads that are in here. It turns out that they get attracted by the optical force. So you have an optical tweezer, essentially, that builds up more and more particles in the structure. So these kinds of structures, lasers in general, are very nice in terms of using as optical resonators. And of course, there are many different geometries. This would be, for example, a disk laser. So if we make the disk laser smaller and smaller, it turns out that you can make a laser with dimensions less than a micron in size. In fact, you can make a laser with a dimension less than the wavelength that it emits in size. In this case, this emits at 680 nanometers, and it's only 600 nanometers wide. And if you make it too small, the laser typically has problems, but they're mechanical in nature, not optical in nature. And so you can make these kinds of structures. And again, if you dip something like this into a solution, the lasing wavelength will tell you what the surrounding is. So these are just very simple ideas of making refractive index sensors. Why would we want to measure refractive index? Well, for diabetes, for example, you want to measure blood sugar. And so the glucose level can be measured by looking carefully at the refractive index. So we're now in a world where we can build systems where fluidic systems can surround optical systems, and we can measure the fluidic systems with spectroscopic tools. So I'd like to talk a little bit about how these can be integrated. Turns out that the fluidic world has a long history too. So we are now somewhere in between the macroscopic world, where valves are millimeters in size, and the microscopic world, where they are microns in size. And we've built a lot of basic experience in making fluidic systems which are rather complex. And these fluidic systems typically consist of valves. The valves typically have a dead volume of about a picoliter, so you can react and combine materials together with that kind of accuracy, 1 picoliter. And you can build them in this mixer. I like this mixer because it's colorful. We have different food dye that comes in here, blue and red and orange and yellow. And you can mix them all together, and it gives you brown. Okay. So that's, it's a great experiment. But basically, this shows you what the capabilities are. Again, things are aligned together in order to make this. Well, one of the things you can do is you can make the same kind of system into a cell sorter. So you can have cells coming in here. They can sort them depending on their nature. And in fact, you can sort, hundreds of cells in a relatively short amount of time in a small device. This device, in this case, is sitting on a CMOS detector array. So we have a fluid system with cells coming in. We have the CMOS detector chip, which is an imaging chip like the one in your cell phone camera. And we have this kind of thing on top of them. And in fact, this is an image taken from the CMOS detector array of the cell sorter, because the dimensions of the pixels making up the image are smaller than the dimensions of the fluid lines. You can actually see the cells, the channels quite nicely. This is what it looks like. It basically has the fluidics right on top of the imaging system. And you can do many things in this kind of a cell sorter that you cannot do in different cell sorters, where you sort statically. Well, let me talk about medical applications. I promised that I was going to talk about biomedical applications. And so I'm going to talk about this kind of thing, a box that allows us to analyze somebody's health. This is science fiction, because such a box, well, it's starting to exist. People are starting to make it. But in the 1960s, only science fiction people thought of it. And what we want it to be is portable. We want it to be battery operated, and we want it to be fast. So the boxes that we deal with today in the medical world and the pathology labs and so on look like this. This shows you sort of the keyboard, so it gives you a scale. And right now, there are systems that are coming out with limited functionality to do the same kind of things that could be handheld and battery operated. So inside of these, we can build chips. And this is one of these chips that we've built. It's a fluidic chip with about 600 valves. It allows us to perform what's called a sandwich assay or an ELISA assay, in which 10 samples can be analyzed, in this case, for 5 antibodies. And the way it works is like this. We have a chip here, which the pink lines here show you the fluidic lines. The blue lines show you the pneumatic lines. And you can put, you have these fluidic lines intersecting each other in this sort of a square grid. So there are valves that separate all of these lines. These are all little valves. And if we close all of these valves and these valves, we can fluidically introduce some reagent, some antibody, into this line. And then we can put different ones in each of the adjacent lines. And each one of these now, each one of these surfaces has different monoclonal antibodies functionalized on the surface. And then we close these valves and we put in the reagent, in this case, blood sample, blood serum or plasma, into this structure. And then we pump it around. And in this case, the overlap, basically the blood, goes over these patches of functionalized regions that were put in first. And the proteins that we're interested in, cancer markers or other things, will bind selectively to these surfaces of the functionalized chip. And after about 100 times of moving the sample over these patches, we have a pretty good extraction efficiency. And then we can finish the sandwich assay by selectively tagging the proteins of interest. And what you get then, if you look at this under a fluorescence microscope, is you get sort of crosses, where you basically look at the patches of material that were functionalized, and you can look at the intensity here. The fluorescence intensity will tell you what the concentration of the material was that you're looking for. And it turns out that for different materials, these are different antibody reactions, proteins. CRP is C-reactive protein. It's a cardiovascular marker. PSA is a marker for prostate cancer. Ferritin, VEGF, those materials that people are interested in for a blood test. And you can see there's a linear relationship between the intensity that you measure and the concentration that was there. And in fact, you can do this in this kind of a format in a chip. And you can do this on real plasma, so human plasma for all these different materials. In this case, thyroglobulin, VEGF, all these different materials can be measured now with this kind of a system. Out of a single sample, you can measure up to 10 different materials or compositions in the bloodstream, and it allows you to build a panel. The idea really is that what we want to build are disposable systems and inexpensive systems that meet the needs of a real clinical application. So one of the key things was, how can we simplify what I just meant and what I just showed you, which was basically a fluorescence microscope type of system. This is what it looks like. Okay. Well, we can simplify it by using things other than fluorescence to measure whether we have a reaction taking place or not. And so one of the directions was, let's simply use magnetic particles rather than fluorophores to tag the materials that we're interested in. And in this case, now we can build a little circuit that has an inductor on the surface. And that inductor, when you have a magnetic particle go into it, bound by a patch of antibody reaction, it changes in frequency the resonance of the circuit. So this is the frequency shift. And in this case, you can measure, you can sense the presence or absence of a single magnetic particle in this kind of a very compact circuit. So we're using magnetic beads now to do these antibody reactions. And what you notice here is we have an electronic chip, entirely electronic, microfabricated by some silicon fab line. And you have fluidics on top of it. And you have magnetic beads in the fluidics, and you can measure cancer markers. And you can see single magnets change the behavior of the frequency of your inductive coils. Let me give you another example of an integrated biomedical system. This system is what we call a polymerase chain reaction system. Excuse me, I'm recovering from a cold. In this kind of a system, what we're trying to do is measure DNA. We look for DNA. DNA from bacteria or from viruses. If it's an RNA virus, we have to convert the RNA to DNA, but we can look for DNA. And the idea of a polymerase chain reaction is that you can take a sample, a single DNA molecule, and you can amplify the number of molecules by going through a temperature ramping process. And the temperature ramping process, I'll show you in a moment how that works. What you have to do is you have to go up and down in temperature to pretty precise temperatures between 95 degrees and about 55 degrees. And what happens is, we all know DNA is this sort of double helix molecule. And if I schematically paint it like this, there's a bunch of bonds, and there are two single-stranded molecules in here. If we increase our temperature to a high enough temperature, the DNA falls apart, denatures, makes two single-stranded molecules. And then, if we lower the temperature and add an enzyme, we can recreate the original DNA by this enzymatic reaction, by what's called an extension reaction. And now we have two molecules. Now we can go up in temperature again, and we can make those two molecules fall apart. And we can again go to lower temperatures, put our enzyme in, and amplify every time we cycle up and down, and we double the number of DNA molecules in ideal situation. Now, what's interesting is that you can do this over and over and over again, and you can observe this reaction by introducing fluorophores. So when you have this kind of a system and you put a light-emitting diode into it and a detector, while if you put in a material that luminesces only when the DNA is in its proper double helix form, then you can see that you get more light out of it when it's not denatured than when it is denatured. So at this point, you would see light coming out, efficient light emission coming out every time you cycle through. And the intensity of that light emission tells you how many DNA molecules you had. So you can observe the intensity change, and you can observe an E at some point that tells you how many molecules you had at the very beginning. This is the basis of quantitative PCR. Now, we built a system to do this a couple of years ago in my lab. This is what it looked like. It had a heater and a cooling system, a Peltier system, and a lot of heat sinks. And the idea was, let's do this very fast. We built this, and then we took it to people in a virology lab. And we observed that in our PCR system that we built, it was similar in performance to others that you could buy. We looked at different materials, different viruses, hepatitis, AIDS, HIV, herpes simplex, and so on. And now it's evolved into a system that is that you can buy. Now, the interesting thing is that we can do all of this for a cost that is much lower than what was typically the cost of a PCR machine. So in the nowadays, we're trying to build systems where you can do this kind of a test within a system that costs as little as $100 that could be used in the third world for disease diagnostics, where nowadays you only have instruments that cost upwards of $10,000 to do this kind of a test. The interesting evolution is that whereas before we had a Peltier junction, now we just have a relay. And this mechanical relay pushes a cold finger onto the bottom of our sample, and it allows us to cool and heat the sample with a very, very simple, reliable system. This is what it looks like. We basically have a relay. We have a sample holder that's a piece of aluminum sheet, and we move this relay plunger up and down. And every time it hits the sample, it cools down the aluminum. And every time it's removed, we have a heater that goes underneath it. And it allows us to build systems that allow us to cycle the sample at relatively fast rates. Typically, you have to wait for about 90 minutes to do a 30 cycle reaction. The modern machines, the best ones nowadays, go down to about 40 minutes. But in our little system, we can do 30 cycles in about 6 minutes. And in fact, that's not the limit. We can go faster than a typical system because of our geometry. It turns out that when we build a system on a metal slab, the thermal heating and cooling is ideal. And so basically we can heat and cool this really thin area very efficiently. Here I'll show you some of the fastest results. In this case, we heated and cooled the sample within 65 seconds for 30 times. And unfortunately, we didn't amplify the DNA. In fact, we had to slow the system down in order to see amplification because the enzyme was not fast enough to keep up with this reaction. And so we went back and did a number of gel electrophoresis experiments, and we see that right around 100 seconds is where you see real amplification. So we're limited by the chemistry and not so much by the hardware anymore. And it's neat to see that we're limited by the chemistry in a system that costs around $100 or $200. So that's not necessarily the only problem. If you want to do a viral test on somebody, just having a PCR machine doesn't solve everything. It turns out that you have to extract the sample, blood typically or something that you can get from the patient, and you have to prepare it. And unfortunately, the laboratory technicians that are able to prepare this are not found everywhere. And so therefore, there's a big effort in my group now to automate not only the PCR amplification process, but also the entire process from the sample blood to the PCR experiment. So we have to have a filter that's built in. We have to have some sort of a thing that lyses, essentially blenders up the material. We use, again, magnets. We like magnets. If we take the magnets, move them back and forth over a sample which has some glass frit in it, turns out the glass particles don't move, but the magnets do move, and you can shear the sample, the bacteria and whatever else is in the sample very efficiently. You can actually even grind out coffee beans in a system like this. And we intend to combine all of these sample preparation systems on a PC board. Again, we're borrowing from the electronics industry. We're taking PC boards, which are available nowadays with a pretty nice resolution. And we believe we can build very efficient sample preparation cartridges. So maybe in the near future, we'll be able to go to this goal of building little systems that are indeed handheld and automated. Now, let me go one step further. I'd argue that nowadays we have all of these wonderful microfluidic systems. And the biggest problem is, that's what I tell my students, we have built chips that can be in a laboratory, but what we really want is laboratories that are on a chip, okay? And so we sometimes call these systems we've built laboratories on a chip, but they're racks, really racks of control electronics that control them. So one of these chips, this would be a chip that I'm particularly interested in, is shown here. It has about 2,000 valves, and it does what we call a single cell analysis. Well, in this case, we look at single cells, and we perform comparative tests. So we actually don't look at single cells. We look at 32 cells that are identical, ideally, or in some way predictably changed. And we load them in on one side, and then we lyse them. We extract their RNA, and we build their mRNA libraries. We measure them. With, again, PCR, we do amplification. And we try to do all of this on the same chip because we don't want to lose these molecules. And so all of this is done on the same chip. This is not a laboratory on a chip because, indeed, it has lots of electronics, and I'll show you that in a moment. This is how it works, though. This has these 32 different chambers. Each one of them has a single cell in it, not two, not zero, but one cell in it. And it's very difficult to optimize the system to do that. And then you open the valve. This is now the lysis buffer. And you do exactly the same thing to all 32 cells. Okay? So now in this case, it pumps around. This is the pump. Pumps around the sample. And then it goes to the next step, which is extraction of the RNA. And that's shown here. We build our own bead columns. And these are little beads. They're functionalized with material that absorbs the RNA and binds it to the surface. This is how we build the bead columns. And basically, at the end of the day, we have this thing. The chip, as I said, has around 2,000 valves on it. These are controlled by these external lines. And these pneumatic lines go to valves, macroscopic valves. And my students call this the head of Medusa because it really does tell us, it's very difficult to hook them all up. And it turns out we can get useful information out of these devices. But I'm not going to go into that. I'm just going to focus on what this looks like. This is what it looks like. You have these valves on the outside of the chip, and they actuate through these tubes, valves on the inside of the chip. And you have a smiling graduate student or postdoc that sits there and tries to connect them all. And that's what they spend 90% of their time doing. What we really would like to do is remove all of this external hardware. We'd like to build systems that are self-contained. And we've developed two technologies that allow us to do this. One of them uses shape memory alloys. And it turns out shape memory alloys are these wonderful materials that change in length or shape because there's a change in the crystalline structure of the wire. In this case, this would be some wire. And it goes through a phase transformation from austenitic to martensitic phase. And as it does that, it shrinks, or go up and down. As you heat it up, typically you shrink the wire. And as you shrink this wire, if you hold it on both sides, you can compress a fluid channel. In fact, you can see a movie here of a fluid channel here. This is the wire above it. You can see it gets squished by this wire. And so this is a very simple geometric system. You put current through the wire, you resistively heat it, and you shrink it. Another way of actuating things on chip is by using electrolysis. So we can simply have water change to hydrogen and oxygen, and that generates a pressure. So we can actually take some electrode here. This is some platinum electrode pattern in an electrolyte. We put a 2 or 0.1 volt signal on it, and we generate a huge amount of pressure. In fact, we generate 3 atmospheres per second in this kind of system. So we have a pretty fast responding system where we can open and close valves by simply putting electrical signals on the chip. And the idea is, of course, to replace what we typically think of as a fluidic system today with something that just works with a little battery. So that's the idea of building these self-contained chips. I'm going to go back for a moment and talk a little bit about lithography, just because it's one of my favorite things to talk about. And, and then I'll end the talk by talking about implants. So let's talk a little bit about sensors. What can we sense with silicon? It turns out that we've been building small structures that allow us to perform tests by using silicon in the group. And the idea is to build systems where you can have a relatively small conductor and you change the performance of this conductor, the resistance, for example, by changing the number of surface states on the surface and by reacting the surface. So it turns out silicon is a wonderful material. We all know that it's made the semiconductor microchip revolution possible. We know that nowadays you can build these kinds of, etch these kinds of structures. You can make them, in fact, this is a 30 or 40 nanometer wide structure array. I should note that the silicon industry now is at the 32 nanometer node. So they actually can do this over 12-inch wafers. And the idea is that you can take advantage of these features not only for transistors, but also for sensors. So when you take a structure down to its minimum size, we actually pride ourselves in making structures down below 10 nanometers. Here are some 10 nanometer wide structures. Turns out that the silicon starts behaving differently than what it does in the macroscopic world. And in fact, one of the things that you can see, so you can, for example, see a silicon array. This movie doesn't work, unfortunately. You can see that these silicon structures become extremely flexible. And they basically can start bending and actually behave more like rubber than what you would expect bulk silicon to behave like. And so we've actually shown that silicon can have roughly 30% elastic deformation that is recoverable, fully recoverable, if you make the structure small enough. Another thing that you can do is you can start with a relatively small structure, and you can oxidize it. You can oxidize it so that you now decrease the amount of semiconductor that you have in the structure by simply oxidizing for a specific amount of time. So you can start with a structure like this, and you can end with a structure that may be much smaller than the original size of your pillar by simply oxidizing the silicon. In this case, we've oxidized the silicon down to about 3 nanometers in size. And if you look at those kinds of structures, you actually see that they start emitting light. And what's really exciting is that when they emit light, you see a change in the wavelength that is explainable by the quantum confinement of the electrons and the holes in this structure. You see this increase, this shift in the wavelength, in the emission wavelength, as you decrease the size from 4 to 3 to 2 nanometers of your silicon structures. And at the same time as you see this shift, you also see the efficiency at which the light is emitted increase. So silicon, which has typically had in traditionally the major flaw of not being a direct bandgap semiconductor and therefore not emitting light efficiently, can be made to emit light very efficiently if you shrink it down to the nanoscale. In fact, you also have to introduce strain. So there's two things you have to do. You have to shrink it as well as introduce strain. This is very exciting, and it's something that we're just very interested in at the moment. We can then take these oxidized silicon pillars, and we can make contact to the top silicon, and then we can put current through it, and you can see light come out. So we can make a relatively efficient light emitters, we believe, out of silicon. We can at the same time also make structures that look like this. You have a contact, you have the bottom silicon, and you can introduce some metal. So instead of making a MOSFET, which has traditionally, over the last 40 years, fueled the semiconductor world and has looked like this, where there's a source and a drain and a silicon channel underneath a gate, we can build a MOSFET like this. We can have a source and a drain and a semiconductor channel, but now we live in a vertical world. We can make this into a sensor. We can build a system where we can look at this as a transistor that is sensitive to the ions that get absorbed to the surface of the gate. So again, we hope that this fits into this box at some point. Well, what if you don't want a box? What if you just want to implant something, you know? It sort of would be uncomfortable for somebody to implant this thing into you. So if you want to have a sensor that's implanted in you, my advice would be to make it as small as possible, okay? And so people have wandered and worked on building smaller and smaller silicon systems that were autonomous. And over the last 20 years, the term smart dust was coined. The idea was to build systems that incorporate some function, have a power supply, and have some sort of a relaying communication system that allows you to measure, say, the temperature on this side of the room versus that side of the room, or things like that. So these things are called smart dust. So far, they've always been in millimeter sizes. So I would actually call them small pebbles rather than small dust. But now we can build systems that are on the size scale of real dust. In fact, they're hard to find if you drop them. Good luck. You know. So the question is, why would you do this? I think the main reason you want to do this is not so much because it's less comfortable to insert it into a patient, but because it's cheaper. Again, if you can build this at a cost of $5 per square centimeter, you can get a lot more of these little things out of your square centimeter than you can get off these. And you really want to build systems that are redundant. You don't want to have an implant that, you know, decides not to work one day, and then you have to go to another surgery. You really want to have something where you can simply switch from one to the other and say, okay, forget about this one, and go to the next thing. So what we really want to do is build autonomous systems. You also don't want to have it move around. So in the brain, for example, if you put a sensor next to a neuron and you want to observe neural functions, the relative motion of your neuron versus your sensor will depend on the difference in mass, the inertia. If I move around and jiggle my brain, then basically the sensor can be dislodged from where it was before. So the idea is to build the smallest possible system that you can get an energy source, some sample preparation, and some intelligence and data transmission into. I'm going to end the talk by looking at these kinds of systems. Silicon diagnostic chips, I believe, can be made out of silicon and optically powered. Much the same way as solar collectors power solar cells, we can power these systems by using lasers to irradiate at the appropriate wavelengths through the skin and power something inside the body. In fact, there are people here that are experts at doing this. Well, I mean, putting lasers into the body, that is. you can build very small analysis systems already. We can do optical or electronic analysis systems by using some of the tools I've talked about earlier. And you can optically also get the information out. We want to build a free space optical data link from a tiny little silicon chip to the outside world. And I want to put this underneath my watch. I want to have my sensor right underneath there. I want to have my little laser in there. And this is what we're working on today. It doesn't look like this right now, but this was an early rendition. Right now, the device is 30 by 30 microns in size, and it looks more like a pancake than this. But the idea is you have a light source, you have a power supply, and you have some sensing electronics. And this light source, there's lots of challenges. That's part of the reason I'm talking about it here. I think there are lots of people here that may be able to help me with some of these challenges. The challenge, for example, is how do I code the information that I read, some voltage somewhere, in such a way that I can transmit this out without having to worry about whether this thing is perfectly perpendicular to my reader or not. Well, we can make this by standard lithography and then using sacrificial layers to take out the material. We sort of know what wavelength we have to operate at. We need to be somewhere in the near infrared for the powering, because we don't want to have the light gets absorbed by the tissue. We don't want to heat up and boil the cells that we're trying to understand. So we're somewhere between 700 and 900 nanometers in the near infrared, which turns out to be a great wavelength to power silicon at. And you have relatively low absorption loss. You can have a solar collector or a photovoltaic. People have actually made these in the past, and they have pretty linear performance in power versus generated current. Turns out that you can get a decent current out of these things, even with very low powers, with what you get out of solar light. It turns out we can operate at maybe 1,000 times more power, which means we can shrink this kind of device 1,000 times smaller. This is what we're building at the moment. Actually, we're building a number of different photovoltaic systems, and are building these systems with the hope of then powering a pulse width modulation circuit that allows us to take a voltage and convert it to a width of a pulse. Now remember, we want to make this as small as possible, so we have to avoid capacitors, we have to avoid resistors, because these are all big devices that typically take a lot of space. And we want to have the entire device at the moment at 100 by 100 microns in area. We want to measure things at about 10 kilohertz so that you can use them for monitoring neural activities. And the idea is to have some voltage signal with some gain. So the circuit we've developed, and we're actually sending it out right now. This is what it looks like. There's a voltage-controlled oscillator circuit, followed by an analog voltage summer circuit. Then you have a SCHMITT trigger and a laser driver. And you basically take a voltage signal and convert it to a pulse width. This is what it would look like. Pulse width would come out. This is not real data. This is theoretical data developed by a SPICE model. Okay. This is the theoretical data plotted from the SPICE model that shows you that whatever voltage you measure, say a neural activity with, you should be able to see a change in pulse width, and then that tells you what the brain is doing or what that particular neuron is doing. And you can measure it all by pumping it from the outside. So this is the idea. You can build systems like Smart Dust that ultimately solve some problem. And it would be a very specific system to solve a specific problem. But the general idea is you want to use optical pumping and powering and optical data communications because it can be scaled down to the very smallest scales. And for this, we've developed some of the smallest lasers that are electrically driven right now. They are plasmon-assisted photonic crystal lasers, and they look something like this. I'll show you a little bit about them. This is a laser that's unusual. It sits on a piece of gold. It's a gold substrate. It has a photonic crystal semiconductor on top of it to confine the light in the lateral direction. The gold reflects it from the bottom. In this kind of structure, that surprised me, you can get Qs on the order of 2,000 or 3,000. And so that's good enough to get this thing to laze. The device geometry is somewhere around, this would be about 2 micron in size. Okay. And we're building these. These are the individual lasers. This is what they look like. You know, little pedestals, because we have to flip chip bond them on top of our silicon chip. And the applications of this kind of technology range from diabetes to dialysis analysis, blood system, osteoporosis, cancer markers for oncology patients, and so on. So there's lots of need. I mean, we basically hope to take these different technologies that I've talked about and build them, miniaturize them into these individual chips. We think about sensors, think about these silicon nanowire sensors, we think about optical refractive index sensors. Maybe these lasers, the same lasers that are used for communication, could be used to measure the refractive index too. And we can build systems that have some complexity and functionality. So we're getting closer to this original goal of miniaturizing systems for medical applications. And I'm very excited about the prospects of reaching those very small sizes and building systems that are actually inexpensive enough. Oh no, Apple. Inexpensive enough that they can be used for real applications and to reduce the cost of healthcare rather than increase it some more. I'd like to thank you for your attention. And again, I'd like to make sure that you understand that the work that I've shown is not my own work. It's done in collaboration with a fantastic group at Caltech. Well, I mean, there are two answers to this question. The first, inductive coupling, we do have some experience with inductors. And at the smallest scale, if we want to make structures 100 microns and smaller in size, it turns out that you actually, the efficiency of inductive coupling versus optical photovoltaic power generation, it actually is not. Photovoltaic optical systems win out because you need a minimum size. You need to have some sort of a critical coupled system. And for millimeter-sized devices, it makes perfect sense to inductively couple, and that's actually what a lot of people do. I think it's the mode of interest. I think as you shrink these devices, it becomes interesting that the efficiency, power, it's sort of like a transformer where one side is really small. The efficiency with which you can get a power transferred becomes much lower, and once you shrink once you make it really asymmetric. And that's the one answer. And now let me give you the other answer, and that is we know about optics a lot more than we do about inductive systems. So we feel comfortable that the optical solution will work. Now, if we knew more about the inductive coupling, then we may feel the opposite. But certainly, it's not. I would not say it's a closed argument. I would not say that inductive coupling won't work. I think that there may be applications where that's preferred. Yeah, the question is, are microfluidic systems more fragile than electronic circuits? They are in a way. Okay, first of all, let me just compare them. The dimensions typically are much larger in the microfluidic circuits, and the reason for that is just pragmatic. Fluids don't move well through very small orifices, and so making things work fast requires you to make them larger. The typical problems that occur in fluidic systems is if you don't filter the fluids, you clog them up. Or if you apply too high a voltage on a sensor, you generate hydrogen evolution. And so that's the kind of failure mode that you typically find in the fluidic systems. Even though they're larger, they tend to be more fickle. My argument would be that it's probably they're more fickle because we're not used to using them properly. If we put ourselves back at the very beginning of the electronics revolution world when the first ICs came out, I think similar problems occurred in the electronics world where things were a little bit more flaky. But now we've become so accustomed that the electronics is so reliable. I think it's whatever 6 sigma reliability or whatever that means. You know, that's not the case in microfluidics yet. But it's probably because it's at an earlier part of this curve of maturation. No. There's also a materials issue. People typically limit themselves when they do microfluidics to 1 or 2 different materials because they know them. Okay, it turns out that there's a huge world out there of chemistries that could work that people just haven't explored yet. So I would argue that our microfluidic system in our body is pretty robust. Okay, and so if we take that as an example, we can try to build things like it, then we'll probably go far along where systems have to be in the future.
One last question. Yeah, can you just bring up the body that if you are
Yeah. Yeah, that's a very interesting question, and it's one of the questions that originally I was very interested in trying to think about. I think that if you make these devices small enough, so if you make the silicon sensor on the scale of a cell, that there's not that much relative motion if it's embedded in a tissue. Having said that, the biggest problem I'm worried about is not so much the mechanical problem, but it's the fact that the body will reject the system. So you have to coat it with something, or you have to introduce something, some anticoagulant or something like that, in order for it to work properly. So I think that's, I think the surface chemistry is going to be the biggest challenge that we'll see as we start introducing these into real systems.